Oh,
I didn't ask. Well, I didn't get to ask.
better not. Sometimes, okay.
Yeah.
All right. Did I give you your test paper back last time? No? Okay.
What's cold in here?
I know. Especially if it's really hot outside and
Yeah.
you feel more breeze inside there. Okay.
Can I go to the bathroom?
Yeah, quickly. You're gonna start in 5 minutes. Let's get started.
Is anyone else coming?
Yeah. Eric's coming. Amy's coming. Yeah, pretty much everyone's coming today. Missing the bio. Test papers. Yep.
Do I have to wear the mask?
Okay, for today's class, the resources are on the online, so you guys can also download that. Let me just quickly update this. Hey, are you free tomorrow?
Not tomorrow.
Are you free next Monday?
Pardon?
Are you free next Monday?
Next Monday, no.
Are you free next Tuesday?
No.
Well, are you free?
Thursday after school.
You go to school on Thursday after 12.
Wait, what?
Aren't you going back to school on Thursday?
I'm like after school.
After school.
Yeah.
Come on Thursday for the test. Okay. Hayden. Hello. Eric. And Hayden. Good. Ashley. No name. Liana. No name. No. Eric. Done. Hang on. Retest. When can you come back? So Hayden, next Thursday, 1st of February.
Come straight after school.
Yep. Come with Haley, because she's coming on Thursday too.
What time is it?
She's have a class at 4:30, so well, if she comes, I don't know, she comes here like 3:40ish, straight after school. I mean, like, what's the point of wasting your time? Just come early, study that your test, and yeah, let's get it done early. Make sure you revise it, make sure you study it, and the questions were pretty, I should say, straightforward, to be honest.
As we did last week, right?
Yeah. So today we are going to do, we're going to cover some of the chapter 5 and then going to focus more on to the scientific skills in biology, that including, you know, because you may need to write on a report about some of the topics. So I give you some introductions to how to write your reports, and those who have done writing report before in the SRP or any sort of the research scientific reports, it'll be a good revision for you too. Okay. Compared to other science subjects, biology takes a huge portion of the scientific skills in terms of the graphing, in terms of data processing, and there'll be lots of readings that you have to go through. So it's something that we're going to practice it again too, and again, we're gonna discuss about validity, reliability, and accuracy as we go too. Okay, and you gotta get quite a thick, fat homeworks to do related to scientific skills today. Okay, retest. Tomorrow, 9.
Teacher.
Hmm?
Teacher.
Monday. You're coming on Monday, anyways.
Chemistry test.
So,
Oh. Wednesday or Thursday.
thank you. Yep, take that. That's all yours then. Wednesday.
Or Thursday.
Pick one. It's either the end Wednesday. You don't come both. Wednesday. If you're coming on Wednesday, you can come in the morning. It's like the last day of your holiday, so you can come here like, I don't know, 12 maybe.
Wednesday.
What time?
So,
12. 12 p.m. Those of you taking chemistry as well, oh, except you, Hayden. There will be a test for chemistry next week. So I said it'll be Monday or Saturday. So for you, Saturday actually. Monday. So we gotta finish the module 1. I'm kind of postponed to finish the module 1 for biology. It takes lots of the information in it too, so I gotta take it divided into 2 parts of the cell requirements, and we're gonna finish some portion of the cell requirements, and I'm gonna finalize that next week too. So please, guys, please bring this worksheet next week. We're going to finish everything by next week. We're gonna get the extra worksheets to do from the scientific skills, and then we're gonna go through the questions together. All right. The results are back. You got your result from your the last week's test. Questions were pretty straightforward, I must say. Okay, it requires a bit of like you need to memorize quite a bit from like exocytes, endocytes, and all the biological definitions too. So we're still doing the cells. So let's I give you a summary of what we're doing in the module 1 about the cells. So let's talk about what we have learning in the cells now. Okay? Hey, what do we learn in module 1? Let's coming back to thinking about from chapter 1 and to chapter 4 prior to chapter 5. Just throw me anything. What do we learn about cells?
Like plants and animals.
So let's do the mind grab. Cells in the middle there. Yep.
Like plants and animal cells.
Plants and animals. So we talked about the different types of the cells. We had a plant, for example, plant versus the animals. Is definitely we did that. Good. Okay. Eric, what do we learn about cells?
The organelles and parts.
Organelles. Okay. Cell organelles. Can you give me 2 examples of organelles?
The ribosomes.
Ribosomes,
And the Golgi body.
Golgi body. Also like a nucleus, and those everything that we have in the cell, inside the cell. What we have inside the cell? What about outside of the cell? We also listen about cell membrane. Okay, we spent quite a lot of time about talking about cell membranes. You have to remember that the phospholipid layers, cell membrane structures and functions of that too. Okay, actually, what else?
That all living things are made up of cells.
Okay, all living things. So we have a thinking about the purpose of the cells too. Can you elaborate a little more?
Like,
Mm. What about this? Did we learn about the difference between eukaryotic versus the prokaryotic? in terms of different type of cells as well too. So comparing the eukaryotic, prokaryotic, like you remember, like the kind of prokaryotic is more like a kind of simple forms too, whereas eukaryotic is more complex. So animal plants will be part of the eukaryotic in that case. Okay, in this case you should be able to draw the diagrams of the eukaryotic and prokaryotic cells, plant and animal cells too. What about technologies? We discussed about different technologies to investigate the cell structures or cell organelles. So that's including the light and the electron, the microscope. That's giving us some scope of looking at how the cell structures too. Okay, let's talk more about the membranes. Inside of within the membranes, that we learned different type of what? Some transport or transportations. We're going to talk more about transportation in module 2, and it's more closely related to your respiratory or the circulatory systems too. But inside of transportations, Eric, what was the 3 different type of the transportations that we discussed? 3. Or maybe 2. If we just divide the transportation into 2 categories,
Oh, active.
well, we have a passive, an active, and the passive. Within the passive, what we have a diffusions, okay, or facilitated diffusions too. We also have the osmosis. And we have an active transportation, so usually requires the energy to transport something to another. So this is basically what we're up to. I mean, we spend quite a lot of time to talk about membranes. I asked you to do the modeling, like, we remember like doing using the lollies and jellies to make the cell membrane like structures, and think discuss about what's that, the limitation of modeling too. Organelles, I've been asking you to memorize all the organelles and functions of the organelles too. It's something that's very important, and also technologies, draw the diagram basically. Okay. So today, we are going to then learn about it here. This is basically giving you the structures of the cells. Okay, thinking about, this is what the structures and functions of cells. The cell has a function of transport and something carrying the very genetic informations. It also has the various types of the cells like this. Today, we're going to learn about cell requirements. Now, requirement means that of the cells is something that closely related to, this is related to the function of the cells. What I mean is, if you understand the functions of the cells or functions of the anything in here, the reason, and you would need like to think about, then to achieve that purpose or to achieve that cell functioning, there are some requirements for that. Okay, so in various requirements, we have a 2. It is generally be a little bit with the materials and about the energy. Cells require specific materials to structure the cells and or like to carry on the informations. Energy during that process is also required too. So 2 categories. You're gonna think of materials and the energies. And within the materials, we're going to think about how we absorb or getting the materials, or how we remove the waste of that too. So in the big categories there, you can think of materials can be made with the 2 parts. Materials or making some composition cells can be inorganic or something be organic. So this is now start point of the chapter 5.1, page 134 there. So technical point, please. The substances needed to be living cells for their functioning are used in 2 main ways. Essential building blocks from which cell and the living tissues are made, and source of store energy for the cells. So just remember that as to achieve the function of the cell, we need to have the building blocks and the energy to move that or connecting those building blocks together. You won't have that because it's from the new worksheet that I gave you today. Okay, so please have the next point too. Organic nutrients are the main supply of the store energy in the living things. but they are used in the structure of the cells, and inorganic nutrients are essential as a structural part of cells and tissues. So let's talk more about the inorganic nutrients or inorganic building block. Okay, so a bit of a summary of the chapter 1, module 1 that we went through. So for the inorganic nutrients, we have a bit something that known as the like we have various examples. For example, water, oxygen, carbon dioxide, nitrogen, and minerals. So how many? 1, 2, 3, 4, 5. 5 are the common inorganic nutrients that we need for the cell functioning. Remember the cell functioning over again and again. Cell requires to have a structure as well as carrying on the genetic informations too. And all that is actually required in the various reasons too. So have a think about the water. Water's making up to what? 70 to 90% of the most organic too. It's an important solvent and transport medium. Highlight that please. So it means that we need to have a water to kind of transport, like a blood, for example, is full of made with the water too. So it's kind of carrying that, kind of making what? Transport that your nutrients that you need. Now, oxygen gas or O2, oxygen is very efficient for the energy supply. We're burning oxygen to produce the energy. Without the oxygen, think about like if you stop your breathing for like even for 1 minute, you will like kind of causing the death or brain death. This is required for the, please highlight this, cellular respiration. Next, carbon dioxide is the ultimate source of the vital carbon atoms for organic molecules, and this is from using the photosynthesis in autotrophs. So autotrophs are green plants. We're going to talk about autotrophs. They're auto means they're automatically producing the energy. Humans are not autotrophs because we are not able to produce the energy by ourselves. We need to consume the food in that reason. Carbon dioxide takes place and leaves as a gas and converted into sugar and eventually returns to the atmosphere. We're going to talk about more about the photosynthesis as we go. Now, nitrogen gas is something also very important because it's the building block of the 20 types of amino acids. Okay, so amino acid is something that's known as the building block, basic building block, the proteins. And we're going to talk about proteins later on too, and why the proteins are so important. But without the nitrogen, you won't get the amino acid, therefore no proteins in your body. Okay. Nitrogen as plant. Okay. Minerals, important to building. Many enzymes and vitamins that are needed for the structure and the functioning. So we need to have some specific minerals. Like the vitamins, think about the reason why we're taking the vitamins every day from the food or taking your tablets, and the various reasons for that. So to fully kind of understand about inorganic nutrients, let's fill it up. The tables in 5.1, page 136 in your workbook or the worksheet that I just gave you today. All right. So we have a few types of the inorganic nutrients. Say, start off with the water that's made with the oxygen and the hydrogen. So where do we get that water from? Okay, going back to the cell structure again too, where do we find the waters in the cells? Any idea? Okay, it's basically made inside of something that's cytoplasm. The big space, taking the space of that. Okay, and the nucleus is also made with a bit of water too. Now, how do we use the water? You just need to remember that this is used as the transport, the medium. It's helping that to transporting the organelles and it's important solvent that we're dissolving something, that the minerals, for examples. Okay, and that's a medium in which all the chemical reactions. It's a medium in the chemical reactions in your body. Let's not talk about what type of chemical reaction we could be involving here, but these are the main functions or main use of the water in the cell's activities. Okay? Mineral salt, for example, chloride, nitrate, phosphate, and carbonates of the sodium, magnesium, calcium, potassium, and ammonium. So, for example, sodium chloride, that's basically what? The table salt. Why do you think we need to consume some of the salt? It's essential. Okay, first of all, it is found in the dissolved in the cytoplasm. Okay, and in the, and do you remember that the vesicule in the plant cells, also in the vesicule in the plant cell. Then, why do we need that? First, this assists all the chemical reaction. It helps to assist helping the chemical reactions. Okay, this is something that we use. For example, sodium chloride assists the water balance, used for the osmosis water balance. In cells, I should say, in cells, and also an essential for the cell membrane functioning. Okay, for example, nerve cells and the muscles are using the sodium and chloride to be activated. It's called the action potential, or sending the signals from that protein the pump of the sodium and chloride. So nerve and muscle cells using electric pulse from that ions. Okay, I'm gonna running out of space right here. Gases that we have the CO2 and O2. Where do we find that from? We find that from they're also dissolved in the protoplasm, cytoplasm in other words. Okay, and think about the way we use where CO2 is known as the main source of the photosynthesis. So that's in the chloroplast. To remember the chloroplast in the plant cells, the green pigment is in helping in responsible of these photosynthesis. The way we use producing the energy in the cells, known as the mitochondria. So again, you should be able to combine all that you learned in cell organelles, the functions of cell organelles, so that we can think about, oh, this is why they require this and that. Okay, for example, mitochondria requires the oxygen gas, so to produce the energy or ATP, known as the cellular respirations. Okay, let's talk about in more details. So CO2, first of all, what do we use CO2 for? That's using that in the photosynthesis. Something that we use in photosynthesis. And then that's also known as products of the aerobic cellular respiration. Again, we're going to talk about more about the energy production, so respiration and photosynthesis later on too. Now, O2 has used for the that's used in cellular, I must say, aerobic cellular respiration. Now, aerobic itself, it contains the meaning of in the presence of the oxygen O2 gas. That's what we mean by the aerobic. Then opposite word would be anaerobic. That's a lack of the oxygen. We can also have the respiration without the oxygen as well too. And this is a product of the Okay, again, we're going to talk more about that, but you can just copy this down. The chemical reaction for the respiration is therefore known as the glucose C6H12O6 plus 6O2, producing that carbon dioxide, CO6, CO2, the gas, plus that to the water, 6H2O, the liquid water, plus the energy, which is known as ATP. We're going to come back to this for the chemical formula. Yes, even though this is a biology class, you need to memorize that right away. This is essential. It's not hard. Easiest way to remember the respiration is, you see that the numbers involved there is always 6 or the multiple of 6. So 6, 12, 6, 6, 6, 6 to remember that. Okay, we're gonna come back to this later. All right, these are known as the inorganic nutrients. Okay. Then how do we differentiate the inorganic to the organic? Any idea? How do we differentiate the organic to the inorganic? Hang on. How do we differentiate the inorganic to organic?
I don't know.
I don't know. Hayden, any thought about organic then to the inorganic?
Like, yes, what they're used for, what they do.
Yeah, or like if you think of like how do we how do we know by looking at that chemical? Oh, this is organic, and how do we know something is inorganic? Not too sure. Okay, so the way we can differentiate organic is generally there's something with the carbons involved. So made with the carbons. So something about chemistry too. Organic chemistry is actually known as carbon chemistry. We're learning about how the carbon structures are made too. The carbons with the various oxygen, nitrogen, hydrogen, or you have the halogens too, kind of combine it all together. So this is something that we have to consider about the organic chemistry or like organic compounds. So organic compounds in the cell's requirements, we have only four, which is carbohydrates, lipids, proteins, and nucleic acid. These are known as organic compounds then. Okay, so let's start off with the carbohydrate. You probably have heard about the carbohydrates quite a lot in the nutrients, the nutritious part. Carbohydrates, which are AKA in short carbs. Okay, we consume carbs every day. I mean, you need to have some carbs. Carbs has from the various sources, but mainly from the grains, rice or flour, from the sugar, and those are known as the carbohydrates. But have a think about here. From carbohydrates, carbo stands for the carbon, hydrate stands for the hydrogen. So from the name of the carbohydrate, we are able to know that, oh, this is something that made with the purely carbon and hydrogen, maybe some of the oxygens too. So it depends on the type of the carbohydrate. We have a few types given here on the table too. We have the mono, so let's start with the monosaccharides. That's the definition here, okay. Mono, let's write it in here, mono, monosaccharides. This is spelling that you can easily get it wrong. That's known as monosaccharides. Monosaccharides. So mono means one. Saccharide means that sugar molecules. Okay, so it simply means saying monosaccharide means we have simply one sugar there. So this is what the simplest carbohydrate. For example, the simplest form of the carbohydrates is simply they're using the sugar. I'm talking about sugar powder you consume. That is known to be the simplest form of the carbohydrate. So they taste sweet, But have a think about the why when we consume too much some sugar, it gives boost up your energy, but that's known as the really active and the fast or the quick, the energy. So it takes a bit of like doesn't require lots of time to be absorbed. It's as soon as you like drink it or consume that sugar, it will kind of absorb in your body, and you can activate the energy too. That's called monosaccharides. Okay, we also have the disaccharide. I'm just kind of placing the space between that to show there's a di means two. So this is made with what? Two sugars. It's a little more the complex sugar molecules. They're made with the two. So as you can see, like disaccharides, we have the maltose, lactose, the milk sugar basically, and the sucrose. You probably heard about lactose from the milk. So some people have the lactose intolerance. They can't digest that in lactose, but that lactose looks like this. It's made with the two conjoined of the two monosaccharides together. Okay, sucrose, cane sugar is also known to be as on the sugar too. So have you do you guys know the difference between white sugar, black sugar, and some brown sugar? I mean, if you go to Woolies, you probably say, oh, we have a brown sugar, we have a black sugar, we have the or if you like to drink the milk tea, the black sugar milk tea. Have you ever heard the black sugar? Have you seen that? Okay. Now, black sugar is basically made from the juice of the sugar cane. That's known as a little more complex sugar than the white sugar. So white sugar is the simplest form. The sucrose, you may think that this is a kind of black sugar from the directly extracted from the sugar cane juice. So that from the process that we go for more process of forming the sugars, producing the sugars, it's getting a more simpler form of the sugar too. We also have something known as polysaccharide. Now, poly stands for many. This means that it has many complex molecules consisting of more than 5. So to highlight this, more than 5 is something that we would say, oh, it's having many of those, up to hundreds. So for example, starch. Starch from your plant is known as the polysaccharides, or known as the complex carbs, carbohydrates. Okay, so if you consume some rice, for example, the bread, that contains the polysaccharides that has the complex carb forms too. So thinking about like converting into the energy from the consuming the food, it takes a bit like a half an hour to the 1 hour to be fully converted into the sugar forms too. Okay, so that is the something we know as the polysaccharides. Starch, cellulose, glycogen is something that we have in there. As an example of those polysaccharides then. So again, what's the main purpose of the carbohydrates? Carbohydrates are always used for your energy source. Okay, and also be maybe there's some structure components. Eric, where can we find the carbohydrates in the cell? In the membrane. What was the purpose of what was the function of the carbohydrate in the cell membranes again?
Energy, structure.
Structure in terms of what? The carbohydrate or the glucose signals receptors are helping to communicate between the cells to cells. Okay, so again, going back to like what we have learned, try to connect them all together so you can draw the big picture of that. That's what I mean by structure components. This is inside of the cell membranes, using them for the cell signaling or the communication between the cells. And we talk about like why it's so important to communicate in between the cells too. If they don't communicate, basically cell will die out because they won't get the sufficient amount of nutrients, or it can get invaded by the pathogens, which is the bacteria or the viruses. Okay, so this is the carbohydrate now. Next is the lipid. Hayden, where do we hear the lipids from in the cell structure again? Give you a hint. point, hydrophobic. Where do we hear? Where did you hear that from?
Like the phospholipid.
Phospholipid of what? Layer of what? What is made of the phospholipid layers? Ashley, what is made of the phospholipid layers?
Like what's in it? Or
What is made of this?
The hydrophobic.
Yeah, so okay, after we're having layers of the phospholipid, what is making the cells? I'm asking really simple. Cell membrane. That's it. Okay, I'm simply cell membrane is made of the phospholipid layers. That's it. That's it. Okay, where am I bringing this one here? Because of lipids over there too. We learned that lipid has the hydrophobic property that you probably see like in a grease on the top of the water because the oil and the water doesn't get mixed together well because due to their chemical properties. You do not need to know why. Okay, that's chemistry. You will do that in module 7 later on. Okay, now for now it kind of gives you an idea that we have a lipid in phospholipid layer and cell membrane. So first thing that gives us that the lipid contains it gives the structure of the cell membrane. This is the first function of the cell membrane, the lipid. Now, lipid is also known as the fats, aka fats. Where do we get the fats? What do we use the fats for? So if you ever seen like go to Macca's and you're ordering some Big Mac, you probably see the energy consumption for that French fries or the chips because of fried foods, okay, in the oil. Now, oil contains the higher energy contents than carbohydrate. In other words, consuming the same amount of the oil and the same amount of the carbohydrate grains, you actually get more energy from the fats. Because this means that it's for the energy storage and usage, but mainly for the storage of the energies in your body. So think about you're getting fat, you're getting more fatty layers. That means that all that unconsumed energy kind of gets converted into the fat forms too, so that you're storing the fats as energy in your body. So lipid contains many carbons, hydrogens and atoms, a few oxygen atoms. The fats and oil in the cells organelle are typically composed of please highlight that triglyceride molecules. So this is known as triglyceride molecules. Tri means 3. We have a 3 glyceride combined. So it's look like this. One of these is look like a glyceride comes here in the diagram. In fact, it has a structure of the, if you want to know in the chemistry, it has a bit of like tails with the carbons. That's known as the glyceride. Okay, again, I'm not going to touch this. I'm not going to talk about too much detail about this. Just need to know that it contains lots of the carbons, hydrogens, and some oxygens. And this is something that's very important. Now, by just looking at the structure of the lipids, carbon, hydrogen, oxygen, you may be questioning, like, isn't that the same as the carbohydrate that we had before? Okay, or the polysaccharides. Now, they have some common similarities, contain the carbon and hydrogen and oxygen, but I'm talking that lipid has lots more dense and quite a lots of the carbon and hydrogen than normal carbohydrate, even for the sucrose or the complex carbs. Okay. So this is the reason why it gives us the energy storage. It's given here, the structure component of the membranes, and also essential structure part of the hormones too. So hormones are made with the fats or the lipids too. We're gonna talk about what the hormones are, like if you ever heard of like a testosterone, okay, estrogen, insulin, for example. Have you heard about the insulin? Okay, that's known as the hormones too. So having not enough fat or the lipid in your body means that you have an improper signaling of your body too. So that's why it's recommended to consume some food. Steroid is a good example. Next part is the protein. Now, protein's also made with the carbon, hydrogen, oxygen, but it has something that's special, that's a nitrogen, and sometimes a sulfur. Combine them, we have something known as amino acids. Now, highlight that please. Amino acids known as the basic building block of the protein. So without the amino acid, it's like a bricks. I've described the amino acid bricks of the building. So you need to have some many bricks to make a building structures. So you need to have many amino acids to make these forms too. So when the amino acids are kind of connected, we call that as peptides or peptide bonds. So the difference between amino acid and peptide is amino acid is kind of individual. Like you can just think that as, oh, we have a buildings, like we have a bricks, okay, individual bricks. That's amino acid. Now, when you start building that, constructing the house, and you built a wall here, just a single wall, that's a peptide. Okay. Now, when you completed your wall, like you made a 4 walls together, then the roof, that's known as the house. That's in protein structure then. Okay, you may think like, oh, this is like a brick, this is like a wall, this is actually a house or building. This is how you can understand about the relation between protein as amino acid to the polypeptide or peptide and to the proteins too. So protein is known to be a little more complex than peptides, or they more like longer than two. They're more like a folded together. So I'm not going to talk too much detail about protein or protein synthesis. That's something you're going to learn in year 12, module 5. But it's good to know that the relation between amino acid to peptide to protein. Now, protein there are about 20 different amino acids that we have. And then that means that we have a 20 different type of the bricks. One could be a white, one could be like the brown, red, black, you name it. Okay, you can just think that out. Means that we can have a various combination between those 20 amino acids, building up to 300 peptide, the polypeptide chains. And we made it from that, from the different shape or the functions of the proteins from it. So protein have many roles. Please highlight that. We can rewrite that here. Proteins. First, it gives you structure. In cells, structure in cells and tissues. Muscles, for example. You consume some protein if you want to grow muscles. Okay. Not only that, it will also be important in the cell membrane. Where can we find this protein cell membranes? What's the function of protein cell membrane? Transport. Or like some other like proteins are heavy in structures too. Do you know? Did we learn about the proteins in the cell membranes? So yeah, we need to have the proteins for the functions, transport, as well as structure of the cell membrane too. Okay. Without that, you won't get good transportations. Some protein they used as enzymes. Highlight that, enzymes. and cells in the hormones and some other things too. But enzyme is the one that made with the protein as well too. We're going to talk about the enzymes more in details next week. Okay, now I hope you found out that I'm talking about like same structures, but depends on that you have a depends on what's making in your body, your tissues, or like some of the parts there. We sometimes need to use carbohydrates, sometimes use lipids, sometimes we use proteins together too. But all of those organic nutrients have a functions of the structural components in your cells. Okay, always related back to the cell membrane as well. Next is known as nucleic acid. Please highlight that. We have 2 types of the nucleic acids in generally. Okay. We have something known as DNA, something known as RNA. These are the 2 popular, or probably the 2 types of the nucleic acid that we have in that too. What's the DNA stands for? It stands for deoxyribonucleic acid. RNA stands for ribonucleic acid. So whenever we have a nucleic acid, it means you have a structure of the nucleotide that look like this. We have a sugar, phosphate, connected to the sugar molecules, and that's made with the nitrogen base. this is known as the basic building block, known as nucleotide. Nucleotide, like a basic building block of the nucleic acid. Now, what is this? Phosphate, sugar, and nitrogen base means it kind of repeated again and again. So it depends how they connected to. We have a different type of the different shape of that. The functions of the nucleic acid. First, every single time we're dealing with the nucleic, think about the nuke stands for something about genetics. So it's all about control the cell. It's a storage of your genetic information, and it's also found in the nucleus too. It's also regulating the cell cycles, that birth and death of cells from it. It also gives that to the so protein synthesis too. Protein synthesis involved. So these are the known, these are the functions known for the nucleic acid. So for example, storage of genetic information is from the DNA. That DNA carries information about you. So taking a cell of you, then we can see like what's the information you have inside there too. Depends on the base, like in the sequence of the bases, then you would have the very distinctive features. So even for like identical twins, they would have a matching 99.99% of the DNA structures, but because of the 0.001%, their two twins are not the same individual. They are a completely separate individual then. Okay, it's quite interesting that having a change of one single base can actually impact you quite significantly too. All right, we're going to talk about the DNA structures, DNA sequences later on in year 12. Don't worry about it too much here, but just try to focus on to the functions and the purpose of having this. Okay. So building block that, I kind of talked about it. Adenine, guanine, cytosine. I will ask you to learn that too. Also cell replications, let's highlight that. This is a part of the cell cycle below, but that's the main function of this. All right, let's talk about DNA versus RNA. A little bit of that, okay? DNA has the 4 bases. That's known as G, T, A, and C. Okay, according to the base pairing rules, what happens to G is matching with the C, A is matching with the T. That's for the DNA structures. So G stands for guanine, cytosine, adenine, thyroxine. Oh, sorry, thiamine, thyroxine is the hormone. Okay. Now for the RNA, instead of having a T, it has GU, AC there, which means G is pairing with the C and A is pairing the U. So U is not a uracil that has the function of that too. Don't worry too much about why it has a uracil, or but this is how we can differentiate the DNA to RNA by looking at the base pairings, and also DNA is something known as double helix structure. As like kind of twisted form of the double helix. RNA doesn't carry the double helix structures at all because it has the various purposes too. We have a tRNA, mRNA, or the RNA. Don't worry too much about it. That's a protein synthesis later on. But that's going to be quite interesting. That how actually we are replicating, how we copying and pasting your cells or the proteins together. All right. In summary, page 42, please. It shows the good summary of all the organic compounds that we have: carbohydrates, lipids, proteins, nucleic acid. As you see, the polysaccharides contain the disaccharides, can be composed of the monosaccharide forms there. Lipid has triglyceride, the fatty acid, and the glycerol. So, good to remember that the simplest form. We have simplest form of the carbohydrate is monosaccharides. The simplest form of the lipid known as glycerols. The simplest form of the protein is amino acid. The simplest form of the nucleic acid is nucleotides. So these are the building blocks, the basic building, or the ingredient that we need to build you. You as in like your whole yourself. You are made with this one in the combination of the inorganic nutrients as well too. Does it make sense? Okay, but it's more about organic compounds that are forming your body shape, and the inorganic comes to like helping that kind of finalizing your shape of your body too. So let's have a go through some questions then. Could you then answer question 1 and question 2, please? We're gonna go through the answers together after that. So give you some templates for organic and inorganic. Organic. In terms of that, let's talk about their compositions then. Okay, the easiest way to figure out organic from inorganic is the presence of the carbon to hydrogen bonding. That is carbon to hydrogen, presence of that. So everything like whenever you have the carbon and hydrogen combinations, we kind of call that as, oh, they're organic. Okay, or they're simply carbons. Now inorganic would be the lack of the carbon to hydrogen then. Now, as you have a little bit of the background about in chemistry, then you would know this is more complex and that inorganic would be more simple in terms of the generally minerals or the elements only. Now examples of those. For the organic compounds, we have many carbohydrates, lipids, proteins or amino acids, proteins for example too, and the nucleic, I must say the protein must be the right category, proteins, and the nucleic acid. Remember that we have 4 of the organic compounds or organic nutrients. For the inorganic, you would have something like calcium ions, or like in the potassium, even for like a sodium ions. What else? We can also have something like what else? Water, sorry, I forgot to talk about water. Okay, and the CO2 would be a good example. Does it make sense? Okay. Now, let's talk about role of those. Where do we use them for? The role of the organic compounds is generally for the crucial for energy production, and also the energy storage, especially the fat, the lipids, and also building cellular structures too. Would be the role of those inorganic compounds or organic like nutrients. Inorganic would be more about physiological processes, that's including that nerve transmissions, transmitting that transmissions, and also be a muscle contraction, sometimes used for the energy productions too. Okay, and also the fluid balance. Having the too concentrated foods or like too salty food is when you want to drink some more water due to the imbalance of the electrolyte or the sodium in your body too. Okay, so that's all we can think about organic, inorganic too. Now, how are you going to distinguish between amino acids to polypeptides to proteins? Something that I gave you in examples. You can think that amino acid as, hang on. Amino acid as?
protein. DNA.
Amino acid as a brick, okay? Peptide, polypeptide would be a portion, like a wall, like a door of your building. Protein would be the whole building itself too. So what's the relationship between them? Okay, you can think amino acid is the basic building block of the proteins. You don't have to worry about the structure of that. And polypeptide is a chain, could be a long or short chain of the amino acids. Lastly, protein are the complex molecules made of more than one polypeptide chains. Again, you can think that as a brick. Give you buildings, okay? Polypeptide would be connecting those all together, making some wall shape with the buildings. Think like here, okay? I can think about a house for the proteins there. Okay, that's how you can think about, visualize the relationship between proteins, polypeptide, and amino acid there. Okay, now question 2, please. Give you about 5 minutes to think about or summarize the some uses of the lipids and describe the use of the lipid in the cells. So A and B. There are many functions of lipids in your body or the cells, but just summarize that into the few. First, it's about energy, storage. We're using fats or the lipids to store your energy. 2, where do we find the lipids and the cells? We're using the phospholipid layers. So it gives you what? Structural component of the cell membrane. Excuse me. That's phospholipid layers of the made with the fat in that case. Now, I'll give you some example there. What do you think about those animals? Like, think of like a little adaptation there. Those animals living in the really cold climate or cold weather. Okay, they tend to building up more fatty layers underneath the skins. Okay, so it's kind of giving us a clue that the reason why they're carrying a lot of fat underneath the skin is there's a good giving that good thermal insulations. The same reason why you're wearing the layers of the coat, layers of the paddings when it's cold too. So think about the penguins, for example. There's a bird, by the way, even though it can't fly. It has a lots of the fatty tissues underneath the skin so that it can protect themselves from the cold weather, okay? Or it can protect them from from the water itself too. So that's something that we would consider. And then also, if you think about the visceral fat, or why you're having the fats in your organs too, your organs kind of wrapped with the fat as well. And that's the reason we need that. If you're running without the fats surrounding your organs, the organs will be kind of collided to each other, can cause the severe damage. So fat is some kind, you can think of kind of packaging or kind of bubble wraps of your organs. So giving that protection of the organs. Not only that, it's also using for making the hormones, so signaling the molecules, like the hormones. So hormone regulations, inflammation, blood clottings, they all requires a bit of the fat as well too. So fat is not, I must say, fat is not that bad. Because some people got a phobia of the fat, of consuming the fats, like from the diets too. I mean, you need to consume some of the fats or the lipids because they have these functions in your body. Okay? That's for the question 2 then. 3. State where in a cell you expect to find the DNA and RNA. Where do you think we can find this DNA RNA? Hey, have a guess. Where do you think we can find this DNAs? The nucleus. DNA, you're defined in the nucleus. Okay, so inside of the nucleus. What about RNA? Where do you think we can find the RNAs from? It'll be found in the various parts, and we can find in the nucleus. Depends on the type of the RNAs you're talking about, mRNA, tRNA, RNA. We can find in the ribosomes. Sometimes we found in the cytoplasm. The RNA, we can find the nucleus known as the mRNA, the messenger RNA. RNA, we can find the ribosome known as rRNA, ribosomal RNA. Cytoplasms or somewhere we can know the tRNA, the RNA in that case. Okay. Again, it's good to know on different type of RNAs, but that's it for now for Module 1, Year 11 state. All right. 4. outline the requirements of the cell. Okay, give you actually 5 minutes to answer or think about the question 4. Have a read that and revise that through. Answer that question. I will ask you each or at least a few for the requirements. You don't have to write this in details, just write in the dot points what's the requirements for the cells. Okay, let's go one by one. Hayden, for the cell requirement, I can categorize into the 2. We need to have some requirement for the structure of the cells. Okay, we need to have for the functions of the cells. Okay, think about here. We have a cell that requires something to maintain its structure. What would be those things that are required for the structure of the cells? Lipid and so we have the organic nutrients. Okay, for example, all nutrients, lipids, proteins, carbohydrates. One more, nucleic acids. These are the main things, also being used for the functions of the energy, producing the energy too, but generally that's used for the structure. Okay, we also need the energy to make the cell to functioning, or to make it functioning as a kind of moving or the living things. Okay, for the energy productions, we need to have the oxygen, because we need to use the oxygen to produce the energy. Okay. Carbohydrate could be related to the energy as we here too, the carbohydrates. It's kind of like using the sugar to making producing that. Lipid can be used to produce all energy storage, production, I should say production, and the storage. Using the various part of that, okay? We can also have the minerals and the ions for the nerve transmissions, using the electrical signals, using the ions. We can have the muscle contraction again. Also, it requires a bit of like a signaling molecule, aka hormones there. Now, there will be many other things too, so let's talk about other factors. The requirements. Now, you may think of like what the cell needs to survive or to be like in the maintain structures. For that, we have many, but we also have the temperature regulations. Cell needs to have some kind of regulating the temperature itself. so that it could be functioning. Okay, so that could be related to functions of that. We can also think about the water. Water is a very essential for the cell structure, as well as we discussed about it's used for the solvent and the transport medium. We can also have the pH balance. pH is what? Acidity or alkalinity of the your levels too. We try to maintain the pH level of 7.2, or like not below the 7, because 7.2, 7.4, or somewhere close to 7, neutralization point. Okay, ion balance and gender materials, we kind of talked about that too. And what else? Lastly, also you need to know about this waste removal. Something that's very, very, very important. Because after we're using that cell organelle, like some of these, they're actually making some byproducts or waste. For example, ammonia is the waste product of your cell respiration or cell metabolism. Carrying that too much like ammonia in your body will be toxic. That will impact on the change in pH, change in water levels. Therefore, it can actually kill you. You may just imagine that you're drinking urine inside the water. With urine contains lots of the ammonia inside that too. Okay, it will be really toxic to drink too much urine. You can drink some if you're in an urgent situation, like there's a bit of things. Okay, and then lastly, we need to have the good communications as well too. Communications. Cells need to communicate to each other. Again, you can relate it back to like the functions of the carbohydrate in the cell membrane too. Okay, so try to give you some simple pictures of the general ideas about cell requirements. So next week we're gonna talk more about this waste removal. I'll probably bring some more notes, extra notes on top of that, because you may need to know the cell functions of the kidney, a little bit of that too. Okay, so waste removal, and we're also going to talk about the enzyme activities next week. So the energy production too. So 3 parts. Any questions up to here? The part 1, cell requirements. Everyone good? Tired? Okay. Let's have a short break, and we're gonna do something else after this.
This.
Yeah. You must be hungry. Yeah. 5 minutes. All right, let's do some worksheets for the scientific skills. Okay, you got the new worksheet now. Let's see what else. So I'll give you some brief summary of the what you need to know for biology 11 and 12. Something that might be a little different to like year 10s. That okay, in year 10 you're probably asked to do something known as the SRP, the student research report, or the project in that case. So in certain stage, any stage of your year 11 and 12, you need to write a scientific report as one of your assessments. Okay, it might be the report, it might be the presentation, depends like how you need to present it. And sometimes we have video recordings, poster, I don't know. Okay, but general like in a general form is the report, and it's good to know how the report is kind of structures. Whenever you're dealing with a scientific report, you will be given something known as inquiry question. That inquiry question is something that we are known as the starting point of the investigations. Okay. Oh, sorry, we're going to talk about this a bit later. Okay. So this is a starting point, and inquiry question is something that giving you that the starting point that think about like a why. It gives you why do you need to investigate an issue. So for example, if you want to have setting up the inquiry question before yourself, you can just question like, okay, I want to know about the relationship between say the pH or the temperature to the photosynthesis. So let's go over it. So you can see that's the worksheet that I give you here. This is giving you some example and giving you the relation between the photosynthesis to the light intensity. So how do we know from that? We see from your data provided, the light intensity is given on your left-hand side and some of the values given here. So as you're questioning yourself, I want to know the relationship between, or I want to know in depth about the photosynthesis. So you set it up in an experiment. So from the inquiry question, it comes to the aim. This is like now why and how you're going to, and what for. Okay, now aim is always be starting with something like this, to something. So it could be like to determine, or it could be to investigate. So that it's giving a clear aim of that, why you're doing that. Make sure you write for example, again, going back to this photosynthesis, you want to investigate the property of the photosynthesis, or you want to investigate, you want to demonstrate, you want to determine the relationship between the light intensity to the photosynthesis. That'll be your aim. The aim needs to be really specific. It's not a vague. We hate to see that vague aim because that doesn't give you anything about the experiment. Okay, after that, after you write your aim, you got to talk about something, safety precautions then. Safety precaution is like, what would be the things that you have to be careful? So this also needs to be specific. For example, if you're dealing with something like an acid, example, like an acid is corrosive. Then you got to talk about how to prevent that risk or how to minimize the risk. Dealing with the acid is something very corrosive, so this is why you're recommended to wear gloves, gowns, closed shoes, safety goggles, known as the personal protective equipments, PPE in short. Okay, do not just write the wear the PPE all the time. I want to know why you need to wear the PPE. Okay, I don't want to know why you need to like performing the risk assessment too. Risk safety precaution is also known as the risk assessment. Now, depends on your writer report too. You may need to provide something like a background research or background information about your topic. So this generally includes the general background informations. Okay, and this also includes in-text citations. Now, in-text citation wise, like you need to provide an author's name and the year it's published. So this depends on schools. Generally, schools follows the either be APA styles. So in-text APA. It could be APA style or the Harvard style. Now, citation can or cannot be maybe a part or part not. Epping boys, 100%. You gotta do the in-text, right? You do. Listen to go. You do too. Okay. Eric, Nominist. You guys don't write the report a lot too, but yeah, sometimes you guys ask to skip this in-text citation. But really, read that in the marking criteria or given like some notification. It will give you that specific format that you need to follow too. Okay, background research. I recommend if you need to write like about 5 pages of report, I recommend to write about a page, at least more than half a page of the background research. This may include some figures, like pictures. Okay, you may show but not too many. Like don't. This is not a PowerPoint presentation. You just need to show some key figures to enhance the understanding about the information that you're providing. Okay, now this is something that you can do. Then what you need to do, you need to show the method. The method needs to be in details. Okay, it also be in the numbers, in number orderings. So step by step, in other words, or the step by step, like a guideline. The easiest way to think about how to write the report and the method is, has anyone seen like the description on the back of your instant noodle cook or the recipe of that? It's been really similar to a recipe of something. That you simply need to follow the recipe or like simply follow the instruction of the cooking the noodles. That's saying 550 milliliters water into the pot, boil that, put the flakes and the noodles in that. Yeah, it needs to be in direct as in like it's kind of ordering that what you need to do. And so that's what happened is if you just follow the instructions, if you follow that kind of manual, then you are able to get the same result as before too. Just like you're cooking noodles in Sydney to Melbourne to Korea, you get the same result because you follow the instruction. Okay, this is why method sometimes come out in your exams too. That you will be asked to write a method for a specific experiment later on. Does it make sense? Okay. Now, after you write the method, you need to show the data or the result. The result sections. Now you need to show the data in tables. Table or tables, and the graphs. Now that graph doesn't need to be in the line of best fit or showing the trend line. Think about when we can show the trend line, where we cannot show the trend line. Okay, if I'm giving you some statistical data saying, hey, I want to know that how many people like in a say right secondary there for biology class. If I collect the data for like your exam marks, okay, you don't see the trend in that. You don't see the relationship between the number of the student to the marks there. What you see there is kind of scattered data or the histogram, saying that some people like the 90s and that, you can't get the trend line or the line of best fit from it. The only time you can show the line of best fit is if you know that x and y axis has a close relationship or some sort of relationship. That's how we describe that as when you're seeing that close relationship between independent and dependent variables, you're able to show in the plotting. So for example, in here you have the light intensity versus the photosynthesis amount over there too. So I want to say that, oh, okay, there will be some relationship between these two. I want to say that some trends of that. Then you'll be able to plot that data and then show in the trend line of it too. Okay, so it depends on the what kind of type of research you're doing. It really depends on you will decide the shape of the graph as well too. Now, finally, discussion. No, second floor. Discussion part. You need to talk about this. You need to assess the validity, reliability, and the accuracy. Second, you need to analyze the result. Analyze result as in like what you be you're using the data of the graph provided or you drew, seeing that or whether you're following to the aim of the experiment, also related to validity later on too. Okay, gonna talk more about validity, reliability, accuracy later on in the chapter here. Okay, then lastly, you need to write the conclusion. That conclude that, oh, this is good. You know, this is successful in testing for the aim. So you are writing the testing for the aim of the experiment. Whether you were successfully tested or not. If not, maybe you can suggest the improvements from it. Maybe some improvements, suggestions. But that suggestion shouldn't go here. It's actually going here, suggestions to improve. Okay, this is a general structure of the scientific report. Okay, in every single time that you need to write a report, those of you studying more than one science subjects, you may need to write 2 scientific reports in every year. Don't worry, in university, you need to write in every semester, like in every term per subjects, at least 4 in 3 months. So by the time I'm in the university, you'll be expert at writing the report. So yeah, hard training. But it's good to have some basics about how to write the scientific report in your high schools too. From year 10, you learn how to write a report briefly from the SRP. Okay, now from year 11 again, let's talk about biology. It is hard to conduct an experiment in the biology. It's kind of limited how much we can do in the biology because sometimes scale is too big or sometimes it's too small. So generally, you maybe do some kind of research on this based on the topics. So that's known as a research task or the depth study in your 11 task 2. Okay, that means you also need to require to write the research report as well too. That's known as the secondary research task. Now, primary is something that you're doing by yourself. You're the one performing the experiment, gather the data, and then write a report about it. So it really depends what you get, but let's have a think about this kind of situation here. First, let's talk about some using the tables. Okay, not this one, sorry. Let's move on to the next page, 5.1. So we have a few, 1, 2, 3, 4, 5, 6 of the report or the topics, scientific topics. Okay, let's have a read that each of those here. So 1, we have a topic 1. Derek wanted to know exactly how the time of the day affects the amount of the photosynthesis done by the plant. He planned to use the oxygen probe in an enclosed environment as an indication of the rate of photosynthesis. That is Derek's design of the experiment there. Okay. 2, Judy wanted to see how the proximity of the artificial light source changed the rate of photosynthesis. She found an experiment online to repeat, which counted the oxygen bubbles produced by pondweed submerged in water with the light of the varied distances. 3, Chris had observed the diffusion of the iodine into a cellulose bag with starch in the class. He remembered that diffusion took a while. He decided to investigate how repeating this step with setup with the different temperatures would affect the time for the contents of the cellulose bag to turn black. Okay. 4. Christie suggested to Chris that you could speed up the diffusion process just by changing the concentration gradient. She planned to demonstrate this by setting up the experiment with the varied concentrations of the iodine solution and comparing the time taken for the contents of the cellulose bag to change color. 5. Talia was unsure what to do with the parts of her microscope. Her teacher told her it was the diaphragm and could be opened and closed to vary the light. Talia decided to play around with the diaphragm position to see how it changed her ability to visualize the onion cells she was observing. The last one, 6. James was curious to see if plants typically found in the shaded environments, such as the rainforest, had different cellular leaf structures to those that with the more sunny habitats. He observed a variety of the prepared slides of the different plant leaves to make comparisons of their cellular structure. Now, all of those 6 experiments, or the design of the experiment is, it started from their thought. So it started from the very specific inquiry question. It's always started from, I want to know that, I'm like curious about this. This is starting for the inquiry question. So for example, Derek, who wanted to know about the time of the day or the photosynthesis, he was questioning about like, I want to know more about the photosynthesis or the property of the photosynthesis. Okay, so he kind of set it up his own experiment, or designed the experiment then, going for the aim after he got an inquiry question. So what would be the aim of the Derek's experiment then? Actually, what would be Derek's aim?
To sort of determine the relationship between
Mm-hmm.
the time of day and
Mm-hmm.
like how much and like photosynthesis.
Yeah, so amount of photosynthesis over here. It's generally like the inquiry question or like a scenario giving you the aim. It's giving you a set aim of the experiment too. But in the aim, as you said, you're able to find that what would be the dependent variable inside of that too. Okay? Then Hayden, from that, Derek's, what would be his independent variable? And what does it mean by independent variable compared to dependent variables?
The thing that changes.
I mean, both of them changes.
Like dependent is what you measure,
What you measure.
and independent is like
Independent is.
thing that changes.
There are both changes, by the way. Both of them changes too. Independent variable is something you're in control of the changing it. Okay, you want to measure something. So to measure the change of the measurements, you change some of these factors. That's called independent variables. So what would be independent variable for Derek's? It would be the time of the day. It would be his independent variables. That he's measuring that it depend on in a different time of the days. That's his independent. Dependent, that would be the amount of photosynthesis. Now, this is where the scientific concept coming out from. Then how are you going to measure that amount of the photosynthesis? Yes, we want to know the relation between the time of the day to the rate of or amount of photosynthesis, but how are you going to measure the amount of photosynthesis then? Eric, how did Derek plan to measure the amount of the photosynthesis then?
The amount of oxygen produced.
Using what?
An oxygen probe.
Oxygen probe. Why would he use oxygen probes?
To see how effective the photosynthesis was.
Because Derek knew that photosynthesis is about the carbon dioxide, CO2, and 6H2O with the light. We produce the glucose and the oxygen gas. Which means Derek already knew that as a product of the photosynthesis, it produced the oxygen. Okay? Where do you think this information needs to go in your report? If you're writing a report about the photosynthesis, this goes on to the background research. Okay? You need to back it up. that, oh, this is why you or Derek chose to the oxygen probe to test for the to determine the amount of photosynthesis. To explain that efficiently, you got to start from what the photosynthesis is, definition of that, providing some chemical equation, maybe a figure, okay, and use of the kind of justify the use of the oxygen probe then. That's why you should put it in your background research then, okay? So it's kind of convincing the reader that, okay, and I kind of get it like why you're using the oxygen probe by reading the background research. Which means, if I'm marking your report too, if I don't get convinced by your background research, then if I don't convince why you're using that specific tools, you gotta mark deducted in every single time. So it's all about the logics. It's all about convincing them. Does it make sense? Okay. So, what about the controlled variables? So let's talk about independent, dependent control variables for each of those. Next page. All right, so we have a different type of variables that we have to use. And remember that this is well used again and again to explain the discussion parts later on. So let's highlight that. First, let's mix and match some independent, dependent control variables that are here, okay? Constant variables, so you can just take this example, the controlled variables. It's the same thing, okay? Something that are constant or control variables. This is something that will not vary at all throughout the experiment. I mean, you do not change that. You do not change, like for example, oxygen probe or the measurement. Okay? Now, if you change that deliberately, like you're like the one in charge of changing something, that's known as independent variable. So it's changed deliberately in the experiment design. Now, when you're measuring that from that change in independent variable, that's known as dependent variable. Okay? Control variable and constant variable are kept identical, which is the same thing here. And what do you know? Independent and dependent variables. Okay, independent variable will vary, will not remain the same. But dependent variables depends on that. Maybe the reaction kind of stopped, then you may get the same dependent variable. So I wouldn't say that's it's a varying every single time. Depends. For example, if you have a relation of the graph like this, I can say, oh, this part is no change at all. Okay, so that's something we can determine from the dependent variable in this case. Then for the experiment 1, investigation 1, can you find it out? What's independent, dependent control variables? And let's do all that, 1, 2, 3, 4, 5, 6 there. I give you an example for 1, investigation 1. Okay, let's go independent variable for this. What's given as time of the day. Okay, dependent variables. That was given for amount of the oxygen. Produced by plant, or photosynthesis. And control variable or control constant variable, same thing, would be known as, okay, let's think about what things we need to keep constant. First, you gotta keep using the same probe. So probe you used must be kept the same. You don't change the probe every single time you're making a new measurement. What else? What else? What about the plant we're using? The type of plant? Same plant. We have to keep the constant. Okay? And if we can control that, maybe like a different factors like a humidity or the temperature. If you can. They can keep the constant, that'd be great. Air plants, and there are various part of that too. So generally, we try to keep the same experimental environment. In other words, okay, just like you don't perform the experiment one in here, one in this room, one in outside the room. So that we try to do the old experiment or the kind of measurement in the one room or one environment so that we know we can compare that directly. Does it make sense to all? Okay, how to figure out independent, dependent control variable. Now, what is important is you got to think about very specific dependent variable. It's not about amount of photosynthesis. It was actually amount of the oxygen produced by photosynthesis. Okay, this is the key that writing your aim of the experiment to. Does it make sense to all? Okay, now could you answer the question for the investigation 2, 3, 4, 5, 6, 2, please? I give you 5 minutes. so 2, 3, 4, 5, 6. 2, 3, 4, 5, 6. Okay. From you, yes. 2. What would be the independent variable for the experiment 2 or investigation 2?
The proximity of an artificial light source.
That this, okay, artificial proximity, okay. So proximity as in like, can we just rewrite it as kind of distance? Okay, the distance of the artificial light source. Distance of an artificial the light source from where? From the plant. Okay. I'm kind of writing the more specific, like more accurate, exact way. Dependent variable, Eric.
No, oxygen bubbles produced by pond weed submerged in water.
Sure. Okay. How are we gonna measure that? What do we actually measure? Are we gonna measure the oxygen?
A probe.
Yeah. So it'll be actually the number of. We got to actually get a number of the oxygen bubbles. That is actually produced from that the high bubbles from the photosynthesis. Okay, control variable then.
Temperature.
The temperature. Okay, maybe the temperature. Anything else?
The water it's submerged in.
Okay, in terms of water, what?
The type of water.
The type of water and
Concentration.
you mean the amount?
Yeah.
Okay, amount and the type of water. Type of water. I mean, you can use the tap water. You can use the different the distilled water, filtered water. But as long as you kept the constant, that's fine. Okay, and also the source of the light. You kept that constant throughout the experiment. So these are all the control the constant variables then. Hayden, what about investigation 2, 3? Independent variable for that experiment is
The temperature.
the temperature. Good. Dependent variable would it be then?
Time for the cellulose bag to turn black.
Very good. Time for the cellulose bag to turn black. Then control variable,
The same type of bag,
same type of bag. Okay.
the same concentration.
Of what? What concentration of what?
Iodine.
Yeah. So same concentration of the iodine. Is it iodine bag or like it's a starch solution? Well, you can say same iodine and same concentration and the starch bag as well too. Because you only change the temperature. You're not changing any of those substances that you're using inside the two. Type of bag, of course, equipment setup, and for each bags, it'll be the same. But most importantly, you need to keep it the same. Good. Okay. So you, yep, you're done. Independent for the investigation 4.
The amount of iodine concentrate.
Amount. Okay, that's interesting. We have a iodine solutions. Oh wait. How are we going to measure the amount of the iodine solution then? Would it be volume or the concentration? Because those two are quite different things, isn't it? Okay, it's actually the concentration. Concentration of the iodine solutions they're using. Okay, then dependent variables. What about the dependent variable then?
Time for the cellulose bag to turn
Yeah, same as the investigation 3. Okay, time for the cellulose bag to turn black. Good. Then control variables are
Temperature, size and type of bag.
size and type of bag.
The same concentration of the starch bag as well.
Very good. Same concentration of the starch bag. And the same type of bag, size and same type of bag, obviously, yeah. That's 2, okay? Anything else? I mean, that's it, pretty much. What about temperature?
Like
Same temperature too. Temperature as well too. Good. Okay, your turn. Independent variables.
Position of this
Mm-hmm. This is actually tricky. Yep, diaphragm.
Position
Diaphragm.
of diaphragm.
Mm-hmm. Yep, position or extend to the because they want to know like what's the microscope diaphragm to which the microscope the kind of microscope's diaphragm DAI PH diaphragm spelling RIGM is open. Okay, so you want to get that's independent variable. So you don't want to know which is the diaphragm that you need to use. Dependent variables.
Ability to visualize the onion.
Very good. Ability to visualize the onion cells. Then what would be its control variables?
Same magnification.
Same magnifications.
Same location of onion cells.
Okay. Shall we start from same onion cells used? That's one. And also the lightening, the microscope used. You must use the same microscope. And then yes, talk about the same magnification as well too. Good. Let's do investigation 6. Okay, independent variable for that is rainforest. So it'll be the different plant slides, and it's various plant slide observed. Okay, maybe you can choose from different species because we're going to compare that, or you're going to kind of justify the rainforest, so you can just say rainforest versus the kind of what we can the sunny species, or different like plants from the different like plants from the different environment. Dependent variables is then you will see that the structure, the cellular structure in the structures in the leaves. So the control variables will be slightly different. In this case, microscope used must be kept as constant too. Now, what about like a person performing that? You probably have from your personal experience in dealing with the light microscope that it's actually like it depends on who's doing that. Some people are faster, and depends on the techniques they have too. You will see the kind of different results of the image too. So it is very important that you keep one person or keep observing that. The person observing the slide, person observing the slides can also be the control or the constant variable too. Okay, and then you also need to look at part of the leaves that, or leaf, I should say, part of leaf observed as well too. It's like you don't look at the you decide to go to the surface area, some larger surface area of the leaf. You only select that portions. Don't look at the stem of that, or the branch of that, the leaf as well too. Good. Okay, well done. This is 4 working out for 1 to 6, then after we identify the independent dependent variables, it is kind of make more sense to write the hypothesis, therefore the aim as well too. So that's next page. Let's write the aims for each of those. Again, the general formula for the aim is we have to start like to determine, to examine, to measure, to investigate. So let's have a think about the aims and the activity 1 in here. So a general highlight that please. General format for the aim is to determine the effect of the independent variable on the dependent variables. This is like a general format. So using that to write the aim for the investigation 1, you may say to determine the effect of the time of the day on the amount of the oxygen produced by photosynthesis. How about that? Okay, the aim contains all that independent dependent variables and giving you the clear, specific aim of the experiment. So you can do for the next 2. What about 2? Investigation 2. What would be the aim of the investigation 2 then? It would be to determine the distance of the artificial light source from the plant to on the number of the oxygen bubbles from the photosynthesis. Does it make sense to write your aim? Or if you're given an aim, that means that you're able to find the independent dependent variables too. What would be the aim for the investigation 3 then? You may say to determine the effect or the relationship between the temperature on the time for the cells back to turn black. On like in the, again talk about a specific experiment too. What about next one? The relationship to determine the relationship between the concentration of the iodine solution in the time for the cells back to turn black. Done. Okay. Tricky part would be this one here, investigation 5 and 6. It would be to determine the extent to which a microscope diaphragm is open to on the ability to visualize the onion cells. This is kind of tricky, like compared to other like an independent dependent variables, by the way, but it makes sense. Last one, aim for the investigation 6 would be to determine the effect on the plant light observed on the rainforest in the sunny environment on the cellular structures in the leaves. Or you may just say, kind of change that, you may say, to determine the differences of the cellular structures in the leaves in the rainforest and the sunny place too. Does it make sense? Okay, so this is the importance of the writing your independent dependent variable clearly. That gives you the clear aim of the experiment as well too. Now, after we determine the aim, I'm gonna leave that over here too, okay? After you determine the aim, then you're able to set up the hypothesis based on the aim. So that's next page. So please highlight that the general structure for the hypothesis is this. If something independent variable has something to do, then dependent variable will that. Okay, so let's use that for the first experiment there. If the time of day change, but you gotta be specific with the time of the day. If the time of day is like after 2 p.m. or longer, if the time of day that we have more sunlight, then you're expecting the amount of oxygen produced by photosynthesis will be increased. See that? I'm still using that your independent dependent variables, but in the case of the you writing hypothesis, give a scenario. More specific scenario in that too. Don't just say the time of day will have an impact on that. Yes, I want to know what would be that impact, specific impact on that. Make sense? Okay, Eric, what would be your hypothesis on the investigation 2 then?
If the distance of an artificial light source from a plant, then the number of oxygen.
Plant what? Plant what? Distance what?
Huh?
If the distance is increased, then
the number of oxygen
bubbles
decrease.
Yeah, that's your hypothesis then. See what I mean in here? You need to be like give a scenario to give that. What if that happened? Then what would be the consequences of that? Make sense? Hayden, what about yours?
If the temperature decreases,
Yeah.
the time for the cellulose back to turn black will decrease.
Yeah, that's a relationship. That's your hypothesis. Again, you gotta use some specific, like some backup to you for the hypothesis too. That should be written in the background research, by the way. Okay, yours.
If the concentration of iodine solution increases, the time for the cellulose bag to turn black decreases.
Yeah, that's a hypothesis too. So after that, you're just gonna test out whether that hypothesis is correct or not. Good? Yours?
What is diaphragm?
Diaphragm. Which Microsoft diaphragm? Diaphragm basically like from the microscope structures there. But have a think about here. If the extent of which diaphragm is, this is a bit tricky. What about do this one? Let's do this one.
If the plant sites observed in different locations.
Plants lie in different locations. Have a think about it here. This is the tricky part that you can't actually follow the general structure there. You may be like saying like, oh, the cellular structure in the leaves in the rainforest would be, like let's be specific, would be big or small like that, or like having more stoma than the cellular structure in the plants in the sunny environment. Okay, so you don't always follow that, by the way. It depends on what experiment, what type of the variables you're given. Yes, up to investigation 4. This is generally what we use in chemistry, by the way. Okay, we can use that a lot. Biology, we can do this. Okay, this is why I picked the biology on the practice the hardest out of 3 sciences. I mean, there's investigation science too, all of these. So there's combine all the chemistry, biology, and physics too. But does it make sense how to write a good dependent designer experiment and then write it down into forms of the aim and hypothesis, giving you some ideas of how to write them? Then, what about the background research? Okay. As I said, you need to construct the conduct the background research. Okay, highlight that here, please. The background research helps to ensure your experiment design actually fulfills your aim and also helps you interpret your result. So it is usually included in the experiment report as an introduction to allow others to know enough about experimental topics to understand the approach used in the method and to be able to understand the result too. Okay, now just reading that, it doesn't probably come to you. I give you some example of the background research that I had from many years ago. I probably share that for many students when they write on SRP, but from year 10. Okay, there you go. That's what I mean by the background research there. Introduction is different too. Now, from, I forgot which school, she got 99%, or got 1 mark deducted, so close to almost 100% for this report. She wrote a report about forming the bioplastic. You probably have it, like I probably shared that report because it's a model report that I'm always sharing too. This is how much of the background research she did, and I wrote it on there here too. So to explain that use of the bioplastic, she first started from the plastics, what the plastic is, okay, and then move on to what the bioplastics are, and then move on to the synthesis bioplastics, goes up all the way. Now, the reason why she must put like that much of depth of the background research is because she's going to use that definition plastic and bioplastic all over again while she's conducting the experiment, especially in the background, in the discussion part at the back over here. So that's the data, trend line, discussion part, and this is how much she wrote for the report too. Starch, accuracy, and that. Written by year 10, by the way. Yeah, okay. This is the expectation from year 11. At least, not that at least, I'm sorry, okay, about this, okay? You may need to write quite a lot, up to 10 pages of report, or up to like 20 pages, depends. Cumberland last year, and they wrote like 50 pages report. Yeah, 3D modeling, and they did 50 pages. That's a limit. And then, yes, someone actually got like 50 pages done, and you got a full mark. Okay. Yes, I'm giving you some ideas about how to write the report as well too. So I will share some of the sample SRP of the report writing as well. Okay. And the thing is, submit your draft. I will mark it and I kind of correct it and give it back to you as you go. That's something that's very important for year 11 and 12. Okay. Anyways, that's the background research that we need to do. So whether like we have the false statement. Okay, let's have a think about true or false of that. Okay, 5.5. So, explain why the background research aids experiment design. I kind of explained over the top. So let's move on to activity 2 here. Decide whether the following statement about background research are true or false. So first, background research is done following the experiment to explain the result. Is that true? Following the experiment, after the experiment, or before the experiment? It's a background, so we do before, prior, so it's a false. What about background research can assist in developing a suitable method to test the aim? It is true. If you have a suitable, a long and kind of descriptive background research, it's helping to write the better aim. Background research explains the experimental result and any limitations of the results too. It is false. It doesn't really explain any limitations. It just gives you some background. Background research ensures an experiment has constant variables. It doesn't really give you that. All right. Background research can assist the experimentals in performing a hypothesis. Yes, it's helping you a lot to write the better hypothesis. And this is something that is one of the main reasons why you need to write the background research. Lastly, background research is usually included in the introduction. Yes, true. That's what you need to concern about background research is that. Okay. So don't worry about this one over here. Then, choosing our sources, don't worry about this, skip that for now. We have a time, it's not really like writing this yet. Writing a method. Is something that I leave that for your things. And we're step by step for investigate chosen activity 1. But now, just okay. Let's skip that for now because I'm running out of time. I want to discuss about discussion. Next page, please. 5.8. Now, finally, let's talk about the validity, reliability, and the accuracy. This is something that you will get in the exams a lot and investigation as well too. So first, start with the validity. Okay, when you have a questions like, hey, do you know, is this experiment valid? Means that it's question simply asking you, did your experiment should say, were your experiments successful to test the aim? Or you may think about it here, instead of taking a test or to hit the aim, and this is probably like the best diagram to understand the aim. If you're shooting a target, the aim is always in the middle. Okay, if you shoot in the middle, I would say yes, you shoot the middle. So I would say it's your shot is valid because you shoot in the middle. So in this case here, whether you shoot in the middle or not, means that to assess the validity, first thing you need to do, you need to see that is there any systematic error in there or not. Now, system error means that there's errors in the system. There's something wrong with the system. This is probably due to the not calibrating, or lack of calibration, I should say, due to lack of the calibration. You do not calibrate, say, your pH meter, or you may say like you didn't really gonna calibrate your weight, the balance too. So it will like give you a false, means that false reading or false measurements In a case you have the systematic error in your result, then I wouldn't say that's a valid, because if you think about it here, if you're shooting, okay, if your scope or your gun or your archery or your bow is not gonna calibrate it, you wouldn't hit in the middle. Does it make sense? You can't hit the middle with like an uncalibrated tools. So that means that you're out of target. It's always been hitting outside of the midpoint. Does it make sense? So you have to figure out whether you have a systematic error or due to that. Or you may think about, it might be due to the severe or significant measurement error. So you misshoot the target somehow. So it could be a mistake, but if you have too many mistakes in there in the 110 repeat, if you have 5 mistakes out of 10, then I wouldn't say that's a valid experiment. You might need to repeat that again. Okay? Or you may just check for all control variables are kept constant. If you see ever your control variable is not kept constant, I can't say the experiment is valid. So for example, if your control variable is a temperature, okay, you need to keep the same temperature, say 22, 25. Now, aircon broken down somehow, so rising the temperature to 40, then I can't tell that's the valid experiment anymore, because you're going against the control variables. Does it make sense? So the way you can think about the target is, this is invalid, but this is valid. Can you see that the valid experiment is always hitting the middle in the target? Okay, or like a testing for the aim. Accuracy. How do we test for the accuracy? This is all about where your measurement, the accurate or accurately, precisely controlled. So to assess the accuracy, you're looking for any random error. I will only repeat this again. You do not have human error. I will kill you if you write the human error in your answer ever from now on. I hate to see that human error. What do you mean by human error, by the way? Like could be human error. It's done by human. It's not done by animals. It's not. What are you gonna say? If animals that can interrupt it, you're only gonna say animal error. You can't say that. It doesn't make sense. So I just wanted to clarify that you only have 2 types of errors: systematic or random. Generally, what you're saying about human error is like a mistakes. While you're measuring that, it's not your day. You kind of slept, or you kind of measured that kind of wrongly, and you didn't read the meniscus properly. That's the mistakes, and that's called the random errors. That happens randomly. But if you repeat that again and again, that's kind of developing into systematic error. What I mean is, you need to read that under the meniscus, the level over here. But if you keep reading the top of that impurposely, that means that's a systematic error. Okay? This is not because of your eyes, because of the mental things probably, or you didn't learn that properly. So random error means that from any random mistakes, from any measurement errors, like whilst you're measuring something, okay, the things happen. Okay, so for example, false reading. Maybe you read the numbers incorrectly too. That's a random error. So mistakes is slightly different to the random errors. Mistake means something that you can't really use in your data, because it's kind of mistake. It's kind of all around. It's kind of screwed basically. You can't use the data. Random errors, you can measure that, so you can see like whether you're actually hitting the target or not too. Does it make sense? Okay. Please remember that 2 errors. Okay, this is something that you can use in chemistry again too, systematic and random errors. Like I'm not talking about biology. This is general scientific skills. The difference between here is mainly due to the systems, mainly due to the faulty like in the machines probably. Okay, maybe like the tools that you're using, the measurement tools. This is the pipette. It's measuring 20 mils, but you realize that this is actually measuring. You get the extra 0.1 milliliter. That is a systematic error then, because there's something wrong with the system. Okay, now, but when you're playing with that, you only draw the, you're supposed to draw up to the line, but you draw only the below that. That's a random error then. Does it make sense? So random is more depends on your techniques, your measurement tools, and your how you deal with the tools. Systematic error is more about the tool itself too. Okay, very important to realize. And this is something common question that we get to. Lastly, reliability. This is like now. Is your data trustable, accountable, or reliable? It's like, can I trust your data? Can I trust your data result? What makes you trust someone? So the way you can think of reliability is always this. To assess the reliability, you only need 2 things. 1, repetition. You need to repeat. You need to repeat the experiment, repetition of the trials, as well as the whole experiment itself too. Preferably, we want to repeat that at least 3 times to increase the reliability. Having 2 may not be enough. Would not be enough to like test like no. The same as this. If you have a friend who's a reliable friend, okay, and saying, oh, I'd say like, oh, Karen is reliable. The only reason I can say he's reliable is he never comes late. Same come at the same time, handle the homeworks, okay, do really well, continuously, at least 3 times in a row. But if I have someone like Hayden, for example, not you, okay, I'm just giving you. If you're coming late, if you're not showing up, if you're not maybe like out of 5 times you only show twice or you're coming 1 late, then I would say, oh, you're reliable. I can't trust you. I don't mean that you're not reliable too. Okay. Same for your friendship too. You say someone's reliable if someone's like always kept the promise, the ride, or they're comes in the same time. It will really punctual to coming in the time as well too. Okay. Same in here. From the many repetitions, you will see that the consistency of the result too. Okay. This is 2 things: consistency and consistency and the repetitions is the only 2 things that we can assess reliability. So it's shown in the targets over here. You will see that there are many reliable data. Okay, due to that, what? High repetitions and high consistency. But this is out of target. So this is the case that we have invalid experiment, but still reliable. Yes, we can have that. Okay, what may cause that? Maybe the shooting scope is kind of broken, so that's what? Systematic error. Does it make sense? Okay. What about this? This is going to have a low validity or invalidity, so you have a bit of validity there. It has a bit of repetition, but I don't see that any consistency within that. That's what? Okay, low reliability then. Okay, now the way I can show the accuracy here is I probably see like, oh, the data's more like in a small dot. Okay, you can just highlight this, you can do that. This is like that high accuracy in terms of target. You just see that the shots are more kind of clumps together in the one spot there. Does it make sense? They're highly accurate. This case over here is a low validity, low reliability. Okay, and if it has some data over here, it also be low accuracy. So the relation between accuracy and reliability in general, when we have a low inaccuracy, it tends to have the low reliability. Because we can't have the reliable data from inaccurate the measurements. Does it make sense? Okay. Now, unreliable. Why is it unreliable? Because you see there's some scatters of the data there. Okay, so it's not consistent. So it's unreliable, low accuracy, or inaccurate, and invalid. This is the case. You have a reliable, valid, and also be accurate too. That you may see, like if the data is more clustered inside that, it's more accurate inside that too. Okay, so you will get lots of questions about assess the validity, accuracy, and reliability. Please remember this. Please try some questions about this. I'm gonna bring some scientific skills questions later on, where we're gonna do the data plottings during the graph of that. So for the next week, please finish up the rest of these. One is finish this, and second is the worksheets. No, homework. Yeah. Finish off this. For homework next week.
Did we finish it?
Yes, activity 2 as well too. Finish activity 2 and finish the homework by next Friday. We're going back to the late night class, 7:30. The school starts next week. Yes.
This one is.
Yeah, activity 2, just activity 2. Yeah, okay. 5.8, let's actually make it here. Activity 1 and 5.8, this page, and this page, next page, and the homework 2. Please try that. I will start with some questions about scientific skills next week. Okay, so this, and yeah, and the page before that, this, that, and the homework. So 2 pages and the homework sheets. Okay, so we're going to continue to talk about those validity and finish up the rest of the chapter module 1 or chapter 5 next week too. And then the week after, module test. I kind of postponed that because I really wanted to cover that before you actually have your data processing. All good? All right. I will see you for your retest as well too. And Liana, if you can come back to the retest, the test, let me know. Well, that's it for today.